Good morning, class 8. Today I am giving some daily practice questions which are important for your final examination. See, question number 1. Name 4 chambers, 4 bulbs, and 4 blood vessels in the human circulatory system. The answer was already written and we have to find the answer from the book. Read the book yourself and find the answer. Name 4 chambers, upper 2 chambers, 2 auricles, 2 ventricles. Left auricle, right auricle, left ventricle, right ventricle. 4 valves, aortic valve, pulmonary valve, etc. 4 blood vessels, vena cava, etc. Question number 2 is as follows. Write advantages of a sexual reproduction B asexual reproduction. Advantages. What are the advantages of sexual reproduction? 2 advantages, and asexual reproduction, right? At least 3 advantages. Question number 3, because you have to practice yourself, so I have given define with examples. Define with examples the following terms. A, see written fission. What do you mean by fission? Write with example. B is written sporulation. C, vegetative propagation. Vegetative propagation. D, budding. E, fragmentation. Regeneration. Regeneration, another systolic pressure and diastolic pressure in human being. Systolic pressure 120, diastolic pressure 80 mmHg. When ventricle contract, when ventricle expand or relax. Another is, see, blood circulation. 2 types of blood circulation, pulmonary and systematic circulation. You have to define systematic circulation. Another, photosynthesis. Define photosynthesis and name 4 raw materials needed. Raw materials needed. Another, there are so many questions, but they are enough. Another is there pericardial fluid. Pericardial fluid. Another most important is, see, reproduction. What do you mean by reproduction? These they are short questions and you have to define. Just like fission, breaking, sporulation, asexual reproduction with spores. Vegetative propagation, asexual reproduction in plants. Budding, appearing and separating an outgrowth from the main part, mother body. Fragmentation, just like fragments, and regeneration, very similar. Breaking, systolic and diastolic pressure, pulmonary and systematic circulation, photosynthesis with raw materials, pericardial fluid, and reproduction. Question number 1 finish, 2 and 3. Question number 3 also finish. Now question number 4. More practice. See, question number 4 is as follows. Write functions of blood. See, one function of platelets, it helps in blood clotting. And one function of WBC, WBC protect us from harmful germs. They are called pathogens. And RBC, right, at least 2 or maximum 3 functions. Question number 5. Here function means all RBC, WBC and platelets. Another is question number 5 is name the diseases caused by overproduction means more, and underproduction is less of red blood cells, white blood cells and platelets. Thrombocyte. See, overproduction of and underproduction of RBC, WBC and platelets. First of all, about RBC. See, red blood cells. Overproduction, polycythemia. Underproduction, anemia. WBC, overproduction, blood cancer or leukemia. Leukemia or blood cancer. And underproduction, leukopenia. Platelets, overproduction, thrombocytosis, and underproduction, thrombopenia. Name diseases. Question number 6. What do you mean by 120 by 80 mm of Hg? You might have heard this word 120 by 80 mm of Hg, blood pressure. People say I am suffering from pressure. Pressure. Is it correct? Do you think I am suffering from pressure? Pressure. That is actually from this point wrong. Don't say I am suffering from pressure. Malai pressure bho, you wrong. Malai high blood pressure bho. You have to say I am suffering from pressure. No. High pressure also no. I am suffering from high blood pressure. High blood pressure bho. What do you mean by 120 by 80 mm of Hg? It means that the systolic pressure is 120 mm of Hg. That means when the ventricles contract, the blood pressure is maximum and it is called systolic pressure. It means the systolic pressure is 120 mm of Hg and the diastolic pressure is 80 mm of Hg when the ventricles relax or expand. Ventricle contract हुँदा blood pressure maximum हुन्छ of माथिको भन्छौँ 120 mm of Hg. Ventricles relax हुँदा blood pressure minimum हुन्छ त्यसलाई 80 mm of Hg भन्छ. Actually 120 by 80 is normal blood pressure. Normal means here normal means healthy. Normal human blood pressure is 120 by 80 mm of Hg, millimeter of. Here Hg means mercury, hydrogen, Latin name or chemical name hydrogen bata Hg bhayeko. Question number 6. Question number 7. What instrument is used to measure human blood pressure? What instrument is used? Sphygmomanometer. This a little bit sphygmomanometer. Sphygmo, you have to sphygmomanometer. Underline. Sphygmomanometer. Say with stress there upon your line. Or sphygmomanometer is used to measure the human blood pressure. See, a sphygmomanometer is used to measure the human blood pressure, which is normally 120 by 80 mm of Hg. If it is 130 or 90, 130 by 90 millimeter of Hg, the person is slightly suffering from high blood pressure. But instead of 120, if it becomes 140, 150, 160, or sometimes 200, 250, and instead of 80, if the person has say 100, 120, 140, I give one example, 150 by 110 mm of Hg, the person is suffering from high blood pressure. But if the person has, instead of 120 by 80, if the person has say 90 by 60 mm of Hg, the person is suffering from low blood pressure. So we have to maintain our blood pressure ourselves. हामीले आफ्नो blood pressure आफैले maintain गर्नुपर्छ. Question number 8. I am giving some questions. Draw a neat diagram of the human heart. Sometimes this question can label 4 parts. Question number 9 is this. Draw neat diagrams to show reproduction in an amoeba, Spirogyra, etc. How they reproduce, you can draw the diagram and see the book and find out the answer. Question number 10. Verify experimentally. Sometimes this question can also be asked. Verify experimentally that oxygen is needed. Sorry, oxygen is evolved. Evolved means produced during photosynthesis. Oxygen is evolved during photosynthesis. See. For the sake of convenience, to make your study easy, I just show from one book. Oxygen is evolved. See, on one book, page number 211, verify experimentally that oxygen is evolved or produced during photosynthesis. If there is photosynthesis, oxygen is produced. What are the things needed? I'll just show you. See, this is the diagram. See, please try yourself. Draw diagram like this. A beaker, hydrilla, glass funnel, water, test tube, oxygen. Photosynthesis and hydrilla plant, funnel. Perform this experiment. After some time, oxygen is produced. And how to test oxygen? It burns more brightly with burning matchstick, or it burns more brightly with magnesium ribbon. See, 2 mg plus O2, 2 mgO. That is the test of oxygen. फोटोसिन्थेसिस हुँदा अक्सिजन निस्किन्छ भन्ने कुरा दुई प्रकारले गर्न सकिन्छ. बर्निङ म्याग्नेसियम रिबन and the burning or matchstick burns more brightly. Question number 11. Question number 11. Prove or verify. Verify experimentally that photosynthesis occurs in a green leaf. It means chlorophyll is needed. I just show you for the sake of convenience. Chlorophyll is needed. See, I show you the diagram. What you take? You take some leaves, boil them in hot water, put 90% ethyl alcohol and iodine solution. केही पातहरूले पातलाई पानीमा उमाल्ने, त्यसमा पहिला इथाइल अल्कोहल राख्ने, त्यसपछि आयोडिन सोल्युसन राख्ने, ब्लू ब्ल्याकमा चेन्ज हुन्छ. टेक सम लिव्स, boil them, put some ethyl alcohol and put some iodine solution. Now the leaf turns blue black. This experiment shows that photosynthesis occurs in a green leaf. That means chlorophyll is needed. If the leaf is not green, suppose the green leaf, you keep the green leaf in a dark room for a long time, and if it has no chlorophyll, the leaf cannot make food. And the green leaf does not turn into blue black. Blue black मा चेन्ज भयो भने यसले ग्लुकोज बनाउँछ, फुड बनाउँछ, नत्र फोटोसिन्थेसिसमा हुँदैन. क्वेशन नम्बर 12. Verify experimentally that photosynthesis occurs in the presence of sunlight. What is the meaning? Sunlight is very important for photosynthesis. No sunlight, no photosynthesis. See, for the sake of convenience, I'll show. See, please draw yourself, which I'll draw. I'll show you. Take a potted plant, pot, any clay pot or any pot, take a potted plant, see, and perform the following experiment. In Nepali, सनलाइट भयो भने फोटोसिन्थेसिस हुन्छ, सनलाइट भएन भने हुँदैन. एउटा पातलाई लिफलाई चाहिँ डार्क रुममा राख्ने, एउटालाई ब्राइटमा राख्ने. डार्क रुममा राखेपछि त्यसमा सनलाइट हुँदैन, त्यसमा फोटोसिन्थेसिस हुँदैन. र ब्राइट रुममा राखेको लिफमा फोटोसिन्थेसिस हुन्छ. टेक 2 leaves, keep one leaf in a dark room for 4-5 days and another room in a bright sunlight. The leaf which is kept in a dark room for a long time does not make food because there is no chlorophyll. It does not get sunlight and it cannot make food. Iodine taste fails or starch taste fails in the leaf which was kept in a dark room for a long time. But the starch taste is successful if the leaf is kept in the presence of sunlight. So sunlight is very essential. If there is no sunlight, what happens? If there is no sunlight, the leaf does not change into blue-black. If there is sunlight, if the leaf is kept in the presence of sunlight, it can make food and it turns into blue-black. That means the starch taste, perform the starch taste. Remember, starch taste. What you do? Take the leaf, boil, put some ethyl alcohol and put some iodine solution. Finally, if the leaf has got sunlight, it turns into blue-black after the starch test. If the leaf was kept in a dark room for a long time, or say 4-5 days, it does not get sunlight and the starch taste fails. Means the leaf does not change into blue-black. In this way, please read this exercise yourself, and I wish you all the best for your BLE exam which is very near. Okay, this was for today.